we have about 20 freezers in our biorepository. 6 full freezers were shipped to the University of Utah about 2 years ago for the SPRINT study. I mean, you can see right here, it says primary serum. The blood was drawn from a patient at a specific time point. The blood was drawn into a tube and spun down. The blood cells flow down to the bottom because they're heavy, and the serum remains on top. You pull that off with a pipette and you put it into a tube, and you can see that it's kind of like a yellowish color, about 500 microliters of serum. And this is why it will take 6 months to pull 35,000 samples, is because you have to be really careful, really slow, really deliberate, make no mistakes, make sure you're pulling the right patient sample, right? And that all takes time. And we want to do it efficiently because Adam and all of the PIs involved in the study are really excited about this cohort of patient samples. And so they want to be able to start getting data back so they can start analyzing it and seeing if their hypotheses are true or not. Serum samples can be stored at -80 and they're pretty stable. Every time you go through a freeze-thaw cycle, those proteins degrade and they start to break down, and we don't want them to thaw. And so we use these little cryopods as a tool to sort samples. The whole chain of custody is going to be just frozen until they get down to Arizona, and then at some point they'll pull those samples, the ones that they want, and they'll thaw them, and then they'll set up the experiments that they need to do down there.